Good morning and welcome. My name is John York. I'm chair of the Department of Biochemistry. Certainly, these are unprecedented COVID times, but despite that, we feel like this is one of the best opportunities and times to train in the sciences, and you have an incredibly bright future. So Vanderbilt is the place to do it, and this program is going to fulfill all of what you have expected. You are here to find your passion and to discover what is worth learning. Our departmental goal is to help you become an expert in critical thinking and scientific inquiry. Character and integrity mean everything during this experience, and you will rise up to all the challenges that present themselves during the next few years. Maybe 2 years, maybe 5 years. I also emphasize that biochemistry makes inclusion intentional, and we welcome all individuals to fulfill their dreams in our diverse and rich training environment. For the next few years, you will immerse yourself in the sciences. If I may use a sports analogy, as I'm a huge baseball fan, I observed that in order to compete, you do not always need to hit home runs. Getting on base is essential. Sometimes you might get a walk, sometimes even hit by a pitch. How about a single or a double? And occasionally swing for the fence. It will provide success. So I want to highlight a few other things about the department. We've recruited a number of new faculty, including 14 new faculty. These typically range in the assistant professor level and a few senior faculty. We have 27 total primary faculty, 4 of which are on the educator track. We have 20 secondary faculty that contribute deeply to our programs and our scientific community, and over 30 research track faculty. Perhaps one of the greatest accomplishments of the department has been Stanley Cohen's work to discover how growth factors control cell division. He won the Nobel Prize in 1986. Our faculty are leaders across the campus. Jennifer Pietenpol is the director of the Cancer Center. Walter Chazin is the director for the Center for Structural Biology. Richard Caprioli is the director of mass spectrometry, and Kevin Schey is director of proteomics. Neil Osheroff is our outstanding director in Academy of Teaching Excellence and Biologic Sciences. Quantitative Systems Biology Center is run by Vito Quaranta. Larry Marnett is our Dean of Basic Sciences. Jennifer Pietenpol is the Executive Vice President of Research at VUMC. And Brant Eichmann is the one of the founders, as well as Brian Bachman, of the Biochemistry undergraduate program. So we have a tremendous breadth of talent within this, and we want to include you in this process. We have field-leading, paradigm-shifting discovery scientists who have invented or led fields. We are incredibly well-funded and have some of the best shared equipment and core resources within the department and within the university. I encourage you to experience all of that. Our labs are newly renovated. We have an incredibly active biochemistry student association. I just want to highlight some of the features of that association. The president is Tyler Hansen. We have a number of officers who contribute to this. We have a seminar and a colloquium. The colloquium is a volunteer basis. You'll get a chance to present your work to a diverse audience of 40 to 50 other students and trainees and postdocs. Before COVID hit, we would meet in person on every other Tuesday, have beer and pizza. We're going to do this virtually through this semester. We have an active seminar series, The Frontiers in Biochemistry. Again, this will be held virtually for this year, as well as we'll have lunches that are boxed for you to be able to pick up before the event. We have a tremendous qualifying exam support group formed by the students. The students also have planned many social events. The Wine and Cheese is perhaps our best attended monthly happy hour. We have coffee and conversations with faculty that our Director of Graduate Studies, Manny Ascano, has pioneered. We have an annual retreat, which is directed by Houra Merrikh and Carlos Lopez. And while this year we've postponed it, we hope to have it in 2021 or perhaps 2022. So this gives you a synopsis. Again, my name is John York. The assistant to the chair is Jennifer Smith. She helps run the department. And as I mentioned, Tyler Hansen is our president of the BSA. And Manny Ascano is our Director of Graduate Studies. Patty Mueller is our coordinator for all graduate students. I just want to again welcome you and thank you for choosing Vanderbilt. You will have an incredible experience here, and I hope you're willing to give us a look. Have a great day. Thank you.
Hi there, my name is Bill Tansey and I study MYC, the emperor of all oncogenes. Why am I interested in MYC? Well, chances are that if you or someone you know has had cancer anywhere in this general area of the body, MYC has been involved. MYC is overexpressed in more than half of all cancers, and it's estimated to cause about 100,000 cancer deaths every year in the United States alone. Now, fundamentally, MYC is a simple protein. It's a sequence-specific transcription factor. It binds chromatin, recruits cofactors, and together they regulate the expression of genes that initiate and maintain the malignant state. Now, time and time again, experimental studies in mice have shown that if you inactivate MYC in the context of an existing cancer, those cancers will regress. So MYC is considered to be a highly validated anti-cancer target. But MYC itself is undruggable. It lacks obvious structure. It doesn't have any of the pockets that medicinal chemists look for when designing drugs. So what this means is that one of the best anti-cancer targets you can think of is also one of the most difficult to target. How can we move forward? Well, this is why we study MYC cofactors, because chromatin cofactors can often be drugged. And we study the chromatin cofactors that we discovered as being important partners in crime for MYC: WDR5, HCF1, and SWitchNEF. And to put it simply, we want to know what they do, how they work, and how we can stop them from working in cancer cells. Now, we use genetics and genome engineering to create sophisticated models that allow us to see how these cofactors work with MYC, or to look at the impact of their loss on cancer cell viability. We use genomic methods to track the location of these proteins on chromatin and probe their role in shaping malignant transcriptional processes. We use proteomics to characterize the interactome of MYC as well as these cofactors to illuminate their mechanism of action. And we collaborate with Steve Fessick to discover drugs that can block coactivator function. We have a robust drug discovery program centered around WDR5, and we're close to picking a clinical candidate, which is incredibly exciting. So if you're interested in cancer or chromatin or drug discovery, and you want to do high impact research in this space, I invite you to rotate with our team. You can find out more by going to our website, tanseylab.com, or you can email me at the address here, and I'd be happy to set up a meeting to talk more. Thanks very much.
Hi everyone, this is Scott Hiebert, and I want to tell you about some really new and very exciting approaches that we're using to look at the dynamic regulation of gene expression in cancer, and really showing how drugs to transcription factors would work if we could develop them. And this was really an approach developed by Christy Stengel in the lab, and together we feel like we can really be great mentors to new students who would be joining. The approach is to use CRISPR to knock in a degradation tag, or actually create a system using CRISPR where we could actually drug transcription factors. What this does is it collapses the time frame from days to just the first hour or so after adding a drug to see changes in gene expression. This really identifies the direct transcriptional targets of transcription factors. We're using RNA-seq, precision nuclear run-on transcription sequencing, which really looks at polymerase dynamics, as well as cut and run. And we do throw in an HA tag so we can look at really robust cut and run patterns of genomic localization. Then we can degrade the protein and watch the protein go away from those genomic sites. In addition, we can add a 3X flag tag and actually use flag mass spec proteomics to really look at all the proteins that are interacting with a given transcription factor. So this is really a comprehensive approach to analyzing transcription factors that are involved in cancer. And we just submitted our first paper. It's under review at Molecular Cell, but we have a series of already tagged genes that are ready to go, and we think we can actually generate papers within 1 year. We have another set of proteins. We've already done the analysis. And what you're going to learn by using this approach is really deep genomics, proteomics, as well as all of the informatics that are used to analyze these proteins. So I hope that you'll consider contacting us. Contact information is on the first slide. Thank you.
Hello, my name is Manny Ascano, and I'm an assistant professor in the Department of Biochemistry and Pathology, Microbiology, and Immunology. I'm also the Director of Graduate Studies for the Department of Biochem, and I would like to extend my warmest welcome to all of you. We all find ourselves in this unique situation, and in some ways it is not an ideal way for us to communicate our science. But I want to encourage all of you to fully embrace this more on-demand approach. Certainly, one advantage is the ability to play and replay the presentations to help you with your decision of finding lab rotations. Günter Blobel, winner of the Nobel Prize in Physiology or Medicine in 1999, was famously quoted in a 2004 New York Times article for stating that if you cannot explain your science to your grandmothers, then you probably don't understand it enough yourself. To me, perhaps the most interesting thing about 2020 is the fact that nearly every human, directly or indirectly, has been impacted by this coronavirus pandemic. That PPE, exponential growth, and RNA virus have found their way into the common vernacular of all languages is amazing. It is fair to say that there was a rapid need for the public at large to educate themselves about the science of viruses, such that yes, our own grandparents have come to understand the gravity of the situation we all find ourselves in. And so it is with this global backdrop with a pre-recorded video that I'd like to share the interests of my lab with you on the biochemistry of host-pathogen interactions and the central players of this stage, viruses. There are over 200 viruses known to cause disease or death in humans, and of these, greater than 70% of them contain RNA genomes like influenza, HIV, and SARS-CoV-2. My lab is interested in understanding the basic mechanisms at play when intracellular proteins make their first contacts with viral nucleic acids, be it DNA or RNA. We are interested in understanding the biochemistry of their interactions. What are the chemical, structural, and sequence features of nucleic acids that lead to their recognition as foreign? We are interested in discovering pioneer host-pathogen interactions. What are the very first cellular proteins that bind to viral RNA or DNA, and what are their functions? Do they act as antiviral restriction factors, or are they hijacked by the virus for proviral means? We are interested in how a cell continues to manage its own gene expression program, which means to transcribe and translate its own set of transcripts, all while trying to suppress the expression of viral transcripts. And finally, we are interested in understanding how the innate immune system of the cell fundamentally understands self from non-self, and what happens when it gets this wrong. So how does my lab attack these very basic questions on innate immunity and host pathogen interactions? We leverage an interdisciplinary combination of molecular biology, biochemistry, chemical biology, high-throughput transcriptomics, and proteomics, along with the computational tools necessary to analyze such high-density datasets. If you are interested in learning more about my lab's work and potential research projects, I encourage you to contact me by email, phone, Twitter, or Zoom. Thank you for your attention, and I look forward to hearing from you.
Hello, my name is Breann. I'm a structural biologist. We study the export of messenger RNA from the nucleus to the cytoplasm. This mRNA export process is essential for the life of all eukaryotic cells. mRNAs have to be exported through the nuclear envelope to the cytoplasm where protein translation occurs. As you can imagine, nuclear export is an essential step in gene expression and is critical for all cellular processes. Our work focuses on two topics. How does the cell prepare nuclear mRNA for export? And how do viruses target this process to suppress the production of antiviral factors? We do both single particle cryo-EM and X-ray crystallography. Just to give you a couple of examples, last year we determined the cryo-EM structure of the core mRNA export machinery, namely the TRAX complex. The role of this complex is to mark mature mRNAs with a licensing factor so they can be exported. This cryo-EM structure for the first time reveals the architecture of this important machinery. We recently also made a breakthrough in understanding how influenza A virus blocks host gene expression. We discovered that the influenza NS1 protein targets the mRNA export licensing factor to block host mRNA export. Now, in a pandemic, you may wonder whether SARS-CoV-2 has anything to do with mRNA export. The answer is yes. We have evidence showing that SARS-CoV-2 blocks mRNA export, and we're trying to determine the structure basis for the relevant protein interactions. If you are interested in our work, just reach out to me and I'm happy to chat.
Hi, I'm Vito Quaranta. I run a cancer systems biology laboratory. We combine experimental and theoretical approaches in an effort to produce a comprehensive understanding of cancer progression and resistance to treatment. We work on how cell identity is produced and how it can be lost in cancer, and on finding treatments that can circumvent the ability of cancer cells to become drug resistant or drug tolerant. Typically, my students publish quite a few papers. Generally, they are authors or co-authors on 3 or more papers, and I see to it that they graduate in about 5 years, no more. They move on to postdocs both in industry and in academic institutions. And in fact, my laboratory is part of a large national consortium of cancer systems biology centers. You can see here we're in good company, which is quite active for meetings of trainees. Now, of course, these meetings are online, but you can still expect to be embedded in a very large community of top laboratories, scientists, and a network of high-level research, meaning that you will have an easy time finding a job after graduation, and high-quality job. So what does a project look like in my laboratory? Well, this is an example where students produced a transcription factor network in a deadly form of lung cancer called small cell lung cancer. And so in doing so, they developed skills to tap databases, so computational skills, and then interrogate these data with bioinformatics tools, with statistics tools. They built mathematical models. They then did computer simulations, to test hypotheses that were generated by these mathematical models. And then they tested these hypotheses experimentally, and with just about any method that you can imagine. We are very broad, the technology that we cover, and especially production of large data sets with high throughput screen systems. So you can expect to be exposed to all of these approaches in my lab and develop some in-depth skills based on the talents you may have or your scientific taste or what intrigues you. So you will become a computational and experimental biologist, which is, as you may know already, a highly valued skill set in the job market today. So finally, let me tell you, I am quite hands-off as a PI. I expect you to take the initiative, develop your own project or projects based on, of course, on the science that is ongoing in the laboratory. So it's your choice, but of course, I'm going to help you and guide you every step of the way. So check out the website. It was in the first slide, and then come rotate in the lab, see what we're doing. Okay, bye. Stay safe.
Hi everyone, my name is Emily Hodges. I'm an assistant professor in the Department of Biochemistry and a member of the Vanderbilt Genetics Institute. My lab studies the role of epigenetics and gene regulation in evolution, development, and disease. Humans share 99% of their genomic sequence, so the full range of phenotypic diversity, like physical and behavioral traits, or risk for disease, cannot be explained by genetic differences alone. And within a single individual, all cell types, no matter how functionally diverse, share one identical genome with a few exceptions. We are especially interested in understanding how these patterns are established during cell fate specification. When cells differentiate from a stem cell to a mature, more functionally specialized cell, each step involves a change in the epigenome. The epigenome reflects how DNA is utilized to direct specific gene expression programs. It is packaged into chromatin via nucleosomes, which influences how transcription factors interact with target DNA sites to influence the how, when, and where of gene expression. Nucleosomes are made of histone proteins that can be biochemically modified to reflect the status of chromatin. DNA itself can be directly modified in the form of cytosine methylation, which has been associated with turning genes on or off. We use comparative functional genomics and bioinformatic approaches to identify and understand gene regulatory diversity between cell types. This involves the use of cutting-edge DNA sequencing technologies that are capable of generating terabytes of DNA sequence data to identify and map locations of these epigenetic features. An example on the left is showing DNA methylation levels for different cell types. By comparing these profiles, we can identify differences that are relevant to gene regulation of a subset of lung cells in this example. On the right, we employed a technique called ATAC-seq to identify changes in chromatin accessibility during a time course of blood cell differentiation. Our research is really at the interface of biochemistry, genetics, and big data science. Projects in my lab also leverage state-of-the-art resources unique to Vanderbilt, like BioView, one of the world's largest biobanks that links genetic information to the electronic health record. We are uniquely positioned to integrate epigenomic information with genotyping and patient data to map and functionally annotate locations of genetic variants associated with disease. Students in my lab are involved in different graduate programs including human genetics, biochemistry, and cancer biology. So please check out our website and follow us on Twitter to learn more, and contact me if you're interested in rotating. Thank you.
Hello. If you're interested in any of the following, the Merrikh Lab may be the right place for you. World domination, stopping evolution, promoting evolution, mutations, anti-evolution drugs, Seinfeld, DNA replication, transcription, conflicts, baby vultures, riboswitches, metabolites, pathogens, pathogen evolution, Neymar, liquid fermented bread, samovars, arguing about where samovars came from, science, Bill Gates, bill from accounting, Texas, French Independence Day, RNA secondary structure, genome structure, gene inversion, gene reversion, 2D gels, 4D mini golf, stocked mini fridges, deep sequencing, shallow sequencing, X-Seattleites, Jeopardy, Dr. Pepper, origins of Merrikh Lab graduate students, Turkish music, Persian music, Russian music, Beyonce. Which bacteria smell better? Organic free-range chicken eggs? International mystery chocolate? Aerialist, Amy Winehouse, Lamarck. Side effects may include groundbreaking ideas, long-lasting friendships, 4-year time to graduation. Email me if you're interested. Thank you.
Hi, my name is Neil Osheroff, and my laboratory works in a class of enzymes known as DNA topoisomerases. These are enzymes that deal with DNA under and overwinding in the cell and take knots and tangles out of the genetic material. Type 2 topoisomerases, which are the ones we normally work on, make a double-stranded break in the DNA backbone, pass another double helix through, and rejoin as though the DNA had never been broken in the first place. Hopefully you can appreciate how powerful these enzymes are because they allow one double helix to simply pass through another, as if the other one didn't exist at all. But the cell pays a very dear price for this because in order to have an enzyme that can do this, it has to make double-stranded breaks in DNA. That's the most dangerous thing you can do to the genetic material. Hence, there's this really tightly regulated balance between cutting DNA that's cut DNA that's ligated. And as a result, you can have drugs that work against these enzymes, because if you have a drug that inhibits the overall catalytic activity, you get slow growth rates, and eventually cells will die in mitotic failure. However, if you have drugs that enhance levels of DNA cleavage, such as a number of anti-cancer drugs, natural products, antibacterials, you basically have drugs that take an essential enzyme and convert it to an enzyme that is a pair of molecular scissors and fragments the genome. As a result, these enzymes are targets for some of the most widely prescribed anti-cancer drugs in the world, like etoposide and doxorubicin, targets for dietary compounds, environmental toxins, and a number of really important antibacterials, including the fluoroquinolones, which are the most widely prescribed antibacterials in the world. We're working with a lot of these drugs. By understanding how the enzyme works and how the drugs interact with them, we have drugs now, by making minor changes, that can overcome resistance and organisms like Mycobacterium tuberculosis. We're working with new drugs such as gepotidacin, which is now in phase 3 trials that make now single-stranded breaks in DNA. And this was Staph aureus, and what you can see here is that we have a structure for it as well. So if you're interested in how enzymes work and how drugs work, please come by and see me, and we'd love to talk to you about maybe coming work for my lab. Thank you.
Hi, I'm James Dewar, and I'm interested in how your genome is copied to completion and what happens when this process goes wrong. Your cells contain 6 billion base pairs of DNA that is replicated with an error rate of less than 1 in a billion. This fidelity is really important, and replication defects cause a variety of diseases. My great passion is in trying to understand how cells completely copy their chromosomes. Now, DNA replication occurs at tens of thousands of locations throughout the genome. At each location, inactive replication machines are loaded onto DNA and then activated to form structures called replication forks, which duplicate the majority of cellular DNA. The final stage in replication is called termination, which occurs when 2 replication forks meet head-on. Now, termination is poorly understood compared to the earlier stages of replication. However, due to my work and the work of others, we're now beginning to understand this enigmatic process. Recently, my lab has discovered that replication forks can become stuck during termination. When this happens, backup proteins are activated to complete DNA synthesis. We're interested in understanding how these backups work and what happens when they go wrong. In addition to termination, my lab is investigating other mechanisms that are important to complete DNA synthesis. In response to chemotherapy, replication forks can get stuck or become damaged, preventing completion of DNA synthesis. My lab is interested in understanding how replication forks are stabilized or restarted so that DNA synthesis can be completed. To study DNA replication, my lab is using 2 complementary approaches. Cell extracts from frog eggs allow us to study DNA replication using the full set of vertebra proteins in a test tube. They're a really good tool for figuring out how things work because we can look at replication with high temporal and spatial resolution. However, extracts aren't cells, so we can't assay for life or death or consequences to cell cycle. For this reason, we're also using human cell culture to investigate how the mechanisms we've discovered impact various aspects of cellular physiology. Our goal is for these 2 approaches to ultimately feed off each other, where discoveries we make using one approach will generate hypotheses we can test using the other approach. If this all sounds like something you might be interested in, please shoot me an email and I'd love to talk to you about potential rotation projects. Thank you.
Welcome to Vanderbilt. My name is David Cortez. I'm professor of biochemistry and director of the basic sciences in the Vanderbilt Ingram Cancer Center. My lab is interested in these 3 numbers. 10,000 trillion. That's the number of cell divisions in a lifetime. 6.8 billion. That's the number of base pairs in our cells that have to be replicated during every cell division. And 25,000. That's the number of DNA lesions that must be repaired every day to maintain genome integrity. So what we're interested in is, how does this work? What happens when it doesn't work properly? And what can we do about it? So my lab approaches these questions using a variety of methods, everything from biochemistry, cell biology, genetics, proteomics, and even structural biology. Just to illustrate some of the recent discoveries from students in my lab, I'll highlight 3. So Thomas was a student who's now a postdoc at Duke who discovered the protein ETAA1 as an ATR kinase activator that controls the cell division cycle in response to DNA damage. Kami Bhatt, who's now a postdoc at Stanford, discovered the RADX protein pictured here that is a regulator of the BRCA1, BRCA2 tumor suppressor pathway to control DNA repair during DNA replication. And Petrie et Thompson, along with Karim Moni, discovered this protein called hemces that actually functions in an entirely new DNA repair pathway that controls responses to the most common type of DNA lesion in our cell, that is abasic sites. And Petrie is now back in the clinic as an MD PhD student, and Karim has taken a faculty position at Mayo. These three examples illustrate the types of studies that we do in the laboratory. We're anxious to have new students and to be happy for you to contact me if you're interested in joining my laboratory. Thank you.
Hello, everyone. I'm Walter Chazin, a professor in the departments of biochemistry and chemistry. I'm also the director of the Center for Structural Biology and the training program in molecular biophysics. My research group, which is shown at the left, is a mix of graduate students, postdocs, postbac students, undergrads, and professional laboratory scientists. We are a diverse and international team because I strongly believe that diversity makes us better scientists. We emphasize inclusion by maintaining a collegial and collaborative work environment. On the right side of this slide is the information to learn more about us and how to contact me. So now that you know a bit about who we are, let's talk about what we do. We are a structural biology laboratory whose research is focused on specific biological and medical problems, not on techniques. Therefore, we use whatever tools are required to solve the problem, including the full range of structural biology and biophysical techniques, as well as in vitro biochemistry and cell-based assays. The lab currently has two broad areas of research: genome propagation and maintenance, and the innate immune response. These have grown out of a personal fascination with macromolecular machines. I want to understand how they function and what causes them to go awry. We isolate specific assemblies and go on to characterize their structure, biochemistry, and cellular functions. I won't be going into great detail in the interest of time, but this slide contains an overview of 4 active projects in the lab, highlighting some of the key systems and the type of questions we're seeking to answer. We're very interested in adding new graduate students to our group, so if you have any questions, please don't hesitate to contact me.
Hi everyone, welcome to Vanderbilt. The Schey Lab is interested in developing mass spectrometry technologies like imaging mass spectrometry and proteomics methods and applying them to study protein aging and protein structure. The tissues of interest are primarily tissues of the ocular eye. Imaging mass spectrometry was developed by Dr. Richard Caprioli, a member of the biochemistry department here at Vanderbilt. Imaging mass spectrometry allows us to create molecular images, that is, spatial distributions of a variety of different molecules within tissues. This includes metabolites, lipids, and proteins. The Schey Lab uses imaging mass spectrometry to study those distributions in tissues of the eye, as shown here. In fact, by the time you're seeing this message, hopefully we'll be funded to create a 3-dimensional molecular map of the human eye. In addition to imaging mass spectrometry, we use proteomics to study protein expression and protein modifications, primarily in the lens of the eye. The reason we focus on the lens is because it's a model system to study protein aging. You see, proteins on the outer shell of the lens are newly synthesized and young proteins, and the proteins in the center of the lens are as old as you are. So if we dissect the lens into regions from outermost cells to the center cells, then we can look at how proteins change with age, including protein expression and how proteins are modified. Recently, Lee Cantrell, a graduate student in my lab, has been able to isolate single fiber cells from the lens and do proteomics analysis to look at protein expression and modification. This is the first case of doing single-cell proteomics on a primary tissue. So I hope these technologies and applications of mass spectrometry interest you. I look forward to seeing you in person in the fall.
Welcome to Vanderbilt. We know it's a strange year, but we're really excited to welcome you to campus where you will receive a great education and opportunities for training. We really hope you'll consider rotating in the Jackson lab. So my lab uncovers the molecular details of the cellular FedEx delivery system, broadly known as membrane trafficking pathways. So it turns out that trafficking pathways and proteins are vital to human physiology and health, and mutations in trafficking genes are linked to a variety of diseases, especially brain diseases like Parkinson's and Alzheimer's. In my group, ultimately we aim to uncover the fundamental principles that govern how cells form structures to move around protein and lipid cargo. We want to understand the molecular basis for disease when these things go wrong. So the lab combines a variety of approaches. For example, we use structural biology techniques like single particle cryo-EM and X-ray crystallography. We use biochemical and biophysical techniques. We're increasingly interested in drug design. We also do cell biology and organismal biology. You'll see here what happens when you knock out some coats in the model organism zebrafish. So Vanderbilt is really a place where you can explore many different things, techniques, areas of biology, even in one lab. And we maintain affiliations with lots of different centers and programs so that all of those opportunities are available to you. So the group itself, we're really very focused on training and mentoring, which I think now are more important than ever. We support our students, we create opportunities for them to flourish, and I think this is reflected in the awards our students win and in the training grants and fellowships that they're awarded. We have many different rotation projects in the lab. They're broadly centered around several different protein complexes. I would love to talk to you about them. Please check out our website, come find us on Twitter, or feel free to email me directly. We have a couple of senior graduate students in the lab right now and 2 postdocs, so the training opportunities are really strong, and we definitely want to recruit some new students as our older students plan to graduate. Please reach out. Welcome to Vanderbilt. We're so glad to have you here.
Hi, my name's Fred Guengerich. I'm a professor in the Department of Biochemistry. I received my own PhD from this department a few years ago. My research involves cytochrome P450 enzymes, particularly those in humans. One of the main interests in our group is the metabolism of drugs, which has relevance in things like inter-individual variations in drug response, drug-drug interactions, and drug toxicity. Our research involves several main lines of investigations. Over the years, our work also led us into work on carcinogen metabolism, the modification of DNA, and mechanisms of how mutations occur and can lead to cancer. Some of the current exciting projects we have in the area include the roles of some DNA polymerases in the formation and processing of DNA protein crosslinks. Here are some of the major approaches in the corner we use in our laboratory for all of these projects. I should emphasize that it's important to be able to think analytically in this lab, and we really emphasize quantitative training. This is a brief introduction to some of the things we do with the 57 human P450 enzymes organized here by substrate class. The xenobiotic substrate P450s work on drugs, being involved in 3/4 of drug metabolism. Today we know that 5 of the P450s do about 90% of the drug metabolism. 4 of these 5, shown with red dots, were first purified in my own laboratory. P450s also really dominate steroid metabolism all the way from cholesterol to estrogens. We are currently working on the mechanisms of several of these enzymes, plus issues relevant to endocrinology. Some of the P450s are what we call orphans in that they don't have a substrate home yet. We have been using some metabolomic mass spectrometry approaches to find substrates and have been able to move some of these out of the orphanage in the past decade. As an example of what we do, consider P450-27C1, which was an orphan until our work in 2015. We now know that it's a retinoid desaturase in fish and changes their visual spectral range during migration and spawning. We humans don't express it in our eyes, but instead in our skin, and 1/4 of our skin retinoids are these desaturated versions. My graduate student Sarah Glass is working on what the function of this reaction is in our skin now. Our lab is open for new students, and so please contact me if you're interested, and I'll be glad to discuss projects, etc. Thank you.
Hello everyone, my name is Breann Brown, and my lab focuses on mechanisms of protein interactions that maintain mitochondrial signaling. When we first learn about mitochondria in school, these organelles are often described as the powerhouse of the cell. However, mitochondrial biology is much more complex, and proper function is indeed critical for overall human health. In fact, it is estimated that around 1 in 5,000 people have some type of genetic mitochondrial disease that can result from a number of factors and present with a range of symptoms. We are interested in understanding how formation of multi-protein complexes, or assemblies, help maintain proper mitochondrial signaling and metabolism. We do this in two major areas. The first is protein assembly controlling production of heme, a critical biomolecule for nearly all organisms. And the second is protein assembly underlying maintenance of mitochondrial DNA copy number. In both cases, alterations in protein assembly can cause severe, sometimes lethal diseases. In our experimental toolkit, we primarily use structural biology, enzymology, and complementary biophysical and biochemical techniques to address how changes in protein assembly and interactions lead to disease. My lab is also a member of the Vanderbilt Center for Structural Biology, which is another great community of researchers that members of the lab interact with regularly. We're located on the 6th floor of Light Hall, so if you're interested in learning more about our research and potential rotation opportunities, please send me an email. Thanks for your time.
My name is Rich Breyer. The Breyer Lab focuses on the pharmacology, physiology, and pathophysiology of prostaglandins, in particular prostaglandin E2. More recently, we focused on the inflammatory processes associated with aging, termed inflammaging. My contact information is shown below. Prostaglandins have long been known to be potent mediators of the acute inflammatory response, a point underscored by the fact that the nonsteroidal anti-inflammatory drugs, or NSAIDs, such as aspirin and ibuprofen, act by blocking prostaglandin synthesis, as shown in the upper left-hand corner of this slide. More recently, it's been appreciated that prostaglandins, in particular PGE2, may also play a role in the low-level chronic inflammation of aging. Breyer Lab has 3 projects ongoing studying the effects of PGE2 in this role. The first one is a project on Alzheimer's disease, which is in collaboration with Fiona Harrison. The second one is a project underscoring the role of PGE2 in hypertension and blood pressure regulation in collaboration with David Harrison. And finally, we have a project on diabetes in collaboration with Maureen Gannon's lab. PGE2 mediates its effects by interacting with 4 e-prostanoid receptors designated EP1 through EP4. These G-protein coupled receptors each bind prostaglandin E2 with similar affinities but signal through distinct pathways. In the case where there's a pro-inflammatory milieu, the pro-inflammatory receptors predominate. Treatment with nonsteroidal anti-inflammatory drugs in young healthy individuals leads to a restoration of balance, as shown in the right-hand portion of this slide. Whereas we predict that in older animals, due to changes in the expression levels of various receptors and the signal transduction pathways that are activated, NSAIDs will no longer be effective. A second separate class of drugs known as inverse agonists that bind these receptors specifically may be effective, however, and we are pursuing the effect of inverse agonists at the EP receptors in treatment of the various metabolic diseases. I'd be happy to discuss any of these projects with interested students at your convenience.
Hey guys, my name is Tina Iverson. Welcome to Vanderbilt. I am thrilled that I hope to be able to meet you at some point. I wanted to tell you a little bit about our laboratory as you are looking for rotation mentors and rotation projects. We use a structure-first approach to look at questions of information coding in several organisms. This involves a number of model systems. For example, we have host-pathogen interactions. How does a host like you know something is a pathogen? Or pathogen-host interactions. How does a pathogen know that you are a delectable host? Traditionally, we have focused on streptococcal infections as our model system, but just in this last year, we have started a few COVID projects that are in their infancy. A main area of research in the lab is information encoding that is interior to you as a mammalian system, and this involves signaling that is downstream from G protein coupled receptors, the largest signaling system in mammals. G protein coupled receptor signaling involves downstream proteins that encode their informational state with a pose or conformation of the protein. And shown here in this morphing movie are two states of a protein called arrestin, going from a basal to an active form. The orange regions in this protein depiction are actually binding sites for other proteins, and you can see that these are changing their shape and creating and destroying binding sites depending on the status of the protein. Finally, we have a number of projects that look at enzymes. Over here to the right, these are often bacterial proteins again, and look at the interface between infectivity and enzyme state, and can involve any enzyme as well as those particularly involved in chemotaxis. I've included a QR code here down at the bottom of the screen that can take you to the lab website if you want to learn more about the research. Within the context of each of these projects, we tend to use a structure-first approach, often X-ray crystallography, but also incorporating NMR spectroscopy and cryo-EM. If you are interested in a rotation project, we can have any of these techniques available for you to learn in the course of your 8 weeks. Again, it was great meeting you. If you're interested in a rotation project, head to the website or drop me an email, tina.iverson@vanderbilt.edu, and I'd be happy to talk to you further. Bye.
Hi everyone, I'm Peck, a new faculty member here at Vanderbilt, and I'm really excited for this opportunity to get to tell you a little bit about my lab today. In addition to my affiliation with the biochemistry department, I'm also a member of the Vanderbilt Center for Bone Biology. My lab's primary goal is to understand the role of the extracellular matrix in musculoskeletal biology. We do this by elucidating the chemical composition of the extracellular matrix in various tissues at a range of developmental stages to map out how the changing cellular microenvironment directs cell signaling and differentiation. We study intervertebral disc formation, spine development, and cartilaginous joint spaces, among others. In addition to understanding the process of normal tissue formation, we're also keenly interested in what happens when the extracellular matrix is dysregulated. With genetic models and injury models, we look at how damage to the cellular microenvironment as a result of genetics, injury, or aging results in disruption to tissue homeostasis. This can manifest in too much bone formation, abnormal bone formation, complete loss of joint spaces, or degeneration of joints. Ultimately, we want to understand these extracellular matrix-mediated molecular mechanisms in health and in disease in order to develop treatment options and regenerative therapies for a variety of musculoskeletal disorders. To answer these scientific questions, you'll use a variety of techniques in the lab. We use a combination of analytical chemistry, biochemistry, and molecular biology, and also musculoskeletal tissue-specific techniques like biomechanical testing, micro-CT, and MRI. As a new faculty member, I'll be able to interact with you one-on-one often. Also, as the first graduate students in my lab, you'll really be on the ground floor of building the lab culture and vision with me. So check out my website for more information on ongoing projects in the lab. And if you're interested in finding out more, please get in touch.
Hi, I'm Will Wan, and this is the introduction to the research in the Wan Lab. Our biological focus is on the negative sense single-stranded RNA viruses. This is a broad order of viruses that include important human pathogens such as measles virus, rabies virus, and Ebola virus. What we're interested in is studying the life cycle of these viruses. And what I mean by life cycle is basically everything they do during an infection. This includes how the virus gets into a cell, its replication inside of a cell, and how it assembles and buds out of the cell. So in order to gain a mechanistic understanding of how the viral machinery accomplishes all these parts of the life cycle, we turn to the methods of structural biology. So what we do is sometimes referred to as in situ structural biology. So in situ is Latin for in place, and when referring to structural biology, essentially what this means is that we're trying to study our biological systems in near native states. For viral life cycle, this means studying intact viruses rather than purifying the individual viral components apart, and studying the viral machinery directly within infected cells. In order to do this, we combine a number of different imaging modalities, including live cell microscopy, fluorescence microscopy, and cryo-electron microscopy. In particular, our method of choice is cryo-electron tomography, a specialized subset of cryo-electron microscopy that allows us to visualize three-dimensional volumes. This is particularly important when we're looking inside complex environments, such as within the cellular milieu, which is often excluded from other structural biology approaches. Not only do we do microscopy, but we also do a lot of image processing with our microscopy data in order to determine high-resolution structures. We're not only users of these methods, but we're also keen on developing new methods. So if you're interested in pursuing work on viruses or a combination of computer programming, image analysis, and microscopy, consider our lab. Our contact details are in the first slide, and feel free to drop me an email.
Hi, this is Chuck Sanders, and welcome to Vanderbilt. We're so pleased that you decided to join us here. So my lab is located over on the 5th floor of the MRB3 building, located in the space of the Center for Structural Biology. So I have about 10 people in my lab, ranging from graduate students to postdocs to staff members. And what we study are membrane proteins. And specifically, we study how defects in membrane proteins cause human disease. And so the proteins or the diseases that we actually look at are Alzheimer's disease, cardiac arrhythmias, and a muscular dystrophy called Charcot-Marie-Tooth disease. And so for all these projects, we use a variety of techniques ranging from our sort of traditional strengths in biochemistry and biophysics, but ranging now into new areas like cell biology and drug discovery. And so thanks for listening, and I'll be seeing you when I lecture you in your bioregulation course, and I hope you have a great day. Bye-bye.
Hi, I'm Bruce Carter. Thank you for your interest in our research. My lab has long studied the regulation of cell death in the developing nervous system. Half of the neurons generated during normal development die as a natural pruning and sculpting process. However, some of these pathways can become abnormally activated in neurodegenerative diseases like Alzheimer's. One area that we've recently become quite interested in is how glial cells regulate neuronal function and survival. For example, in studying myelination by Schwann cells in the peripheral nervous system, which you see here, the Schwann cells are in pink and sensory neurons of the dorsal root ganglia are in green. We found that when the Schwann cells lack a neurotrophin receptor called P75, the Schwann cells are fine, but they have altered lipid metabolism, and this causes sensory neurons to die. This is particularly interesting because altered lipid metabolism in Schwann cells has been found in a variety of neurodegenerative diseases like Charcot-Marie-Tooth disease and diabetic neuropathy. We're now exploring the mechanisms involved in this. Another project where we need to recruit someone is investigating the role of an engulfment receptor in microglia. Several years ago, we discovered a receptor called JEDI, which we like because it's got a fun name. JEDI is involved in the clearance of dead sensory neurons by satellite glia. While studying JEDI knockout mice, we found that it's also expressed in microglia in the brain. Here are some microglial cells, and their phagocytic ability is indicated by the little red beads you see that they've eaten. We found that the Jedi knockout mice have a deficiency in the ability of these microglia to eat dead cells. And one of the outcomes is an alteration in neurogenesis, or the generation of new neurons in adults, which we're now exploring. You can find out more on our website, and I'd be happy to talk with you about any of our projects. Just send me an email.
Hi, my name is Marija Zanic. I'm an assistant professor of cell and developmental biology with secondary appointments in departments of biochemistry as well as chemical and biomolecular engineering. And our lab is fascinated by the dynamic architecture of the microtubule cytoskeleton.
So microtubules are biological polymers that have many important functions inside of cells. They provide networks for intracellular transport. They're absolutely essential for processes such as neuronal development, and they build intricate structures. So this is a structure called the axoneme inside of the flagella and cilia, and allowing this unicellular organism to move. They also build this highly intricate dynamic structure called the mitotic spindle, which ensures proper chromosome segregation during cell division. So to study microtubules and their dynamics, our lab uses a highly interdisciplinary approach. We always start by observations made inside of cells, which allow us to identify key players as well as functions of microtubules. Then by and large, we use the tools of biochemistry and biophysics to recapitulate microtubule behavior outside of cells in highly controlled conditions. We use microfluidics to control the microtubule behavior, and we also use total internal reflection fluorescence microscopy to image microtubules. And then we employ math and physics and computational modeling to capture and recapitulate and model microtubule behavior in silico. So here are some of the examples of the recent research publications from the last 2 years in our lab. Needless to say, we have a highly interdisciplinary team of scientists. We come from diverse scientific backgrounds as well as different cultures. We're a highly international lab. And we have a lot of also different kinds of rotation projects. And we would love to talk to you if you're interested.
Welcome to Vanderbilt.
Greetings, bright young charges. My name is Mimi Blind. I'm the daughter of Ray Blind. My dad is making me record this video to tell you about his research. My dad studies little fat and sugar molecules called inositols, like John York studies. John York gives my mom and dad lots of bourbon when we go trick-or-treating at his house. Reminder, this script was written by Ray Blind. My dad studies how those little fat and sugar molecules activate proteins that bind to DNA using X-rays to study the shape of the proteins. He wants me to say lots of his projects are easy-peasy structural biology. But my dad also has projects that study how the fats and sugars turn on genes. Talk to him about those projects if you are interested in epigenomics. My dad also has computer-only projects where when stupid coronavirus ruins everything again. He has a few of those projects. Slide, please. Here are the people in my dad's lab. Merced is very nice and I am almost as tall as her. Everyone in my dad's lab is nice. That is important if you have to work there for 6 years. I am only 8 years old, so that is a long time. In closing, I, Mimi Blind, encourage you to pick a biochemistry lab. It is the best department because of the wine and cheese parties. With strong leadership and a dynamic faculty with high, innovative, yet feasible projects, one could not wish for a better graduate experience. Peace out, peeps!